Yeah, so I just want to talk about how eutrophication can influence amphibian reproduction in the Cope's gray tree frog. All right, so I'm assuming that most of you have a really good understanding of eutrophication already, so I'm not going to cover too much of the mechanics, but I'll just hop into how eutrophication can be harmful. Let me turn on my laser pointer. All right, so as these nutrients are being pumped into freshwater systems, what we see is this basically unlimited increase in biomass within the water body. And as that biomass increases, we also have an increase in decomposition, which kind of consumes more oxygen than can be produced in that system. And eventually that leads to a crash in oxygen, which affects all of the organisms within that water body, not just amphibians. So fish, invertebrates, macrophytes, everything that relies on that freshwater system can be impacted. So eutrophication can be harmful in a few different ways. We see alterations in community structure, and we see shifts in dominance between species. So if some species has a specific function, like the cattails that we know of in wetlands and things like that, if it moves to something like purple loosestrife, then the function of that ecosystem can really be impacted, and it's not going to do what it's supposed to do in that area anymore. We can see things like shifts from anchored plants to algae and floating plants, which can impact sunlight into the water column, which really impacts productivity below the surface. So we have a really good understanding of the direct effects of eutrophication. For example, these mass mortality events as a result of a complete lack of oxygen. However, in the case of sublethal effects, when eutrophication is not that extreme, there's going to be a lot more indirect effects that we don't understand super well. So when oxygen is reduced rather than completely gone, we're going to see a lot of movement within the water column. Things are going to be migrating among water systems and around, looking for better sources of oxygen. We might see a lot more surfacing. And then in the case of reduced visibility, this is really going to impact things like predator avoidance, foraging, and even breeding behavior. So we know that there's a lot of selection pressures when it comes to habitat location and breeding location. And we know that specifically amphibians have a lot of adaptations accumulated to mitigate a lot of those risks. So things like predator avoidance and oxygen volume available. That goes into those site selections. But when we think of eutrophication as a risk, the question that we want to ask is, do amphibians have the ability to assess eutrophication as a risk and all of the consequences that come with eutrophication? So there's a couple potential trade-offs when it comes to low nutrient versus high nutrient. So when we move into a low nutrient system, there's potentially going to be increased vertebrate predation. There may be more predators, there may be less cover available for larval amphibians. And then with that low nutrient, we're not going to see as much algae or as much plant cover. However, with that less biomass, there's not going to be as much decomposition. So we're probably going to see less CO2 buildup in those systems. So when we move into a higher nutrient system, we're going to see a lot more algae and a lot more plant cover, which might translate to more food availability for those larval amphibians. And then for a time, we're going to see an increased amount of dissolved oxygen within that system. And that might also mean less invertebrate predation, whether it means just more cover or if that high nutrient system is not as attractive to those invertebrate predators. So going into this experiment, I had two main objectives. The first being to investigate how adult amphibians respond to eutrophication during their breeding. For this, I used Hyla chrysoscelis, or the Cope's gray tree frog. It's really abundant in Kentucky, and their breeding behaviors are really easily observable. So I was hoping to follow up that half of the experiment and determine if that breeding behavior is adaptive for larval survival. So we wanted to see if, the parrots were making the correct choice. Unfortunately, I did not get to complete that experiment. I had it all started and ready to go, but unfortunately I added the wrong tadpoles to all of my tanks. And then I had hundreds of backup tadpoles that were the correct species, and they were all eaten by predaceous bugs. So that one was kind of a bust. But moving on to the experiment that I did get to do. So I used 300-gallon cattle tanks as my experimental ponds. 15 were designated as a treatment, and then 15 were the control. All 30 of my tanks received just standard amounts of leaf litter, macroinvertebrate-rich substrate, and the same amount of algae. And then my treatment tanks received a treatment of fertilizer. I just used Osmocote. It's really popular in these mesocosm eutrophication studies. And then those were left to sit for about 25 days before any sampling occurred. So once that 25-day period was over, I started doing nightly surveys of male activity. So I just went out and looked for any males that were calling on any of those tanks. And then the morning after, I would go out and collect any eggs that were laid in those tanks. And that is what those eggs would look like. Obviously, it's really difficult to see if they're in duckweed. Okay, so after I got those numbers, I used a chi-square analysis just to detect if there were any differences in male and female preference between the treatment and control tanks. All right, so just getting this started to make sure everything was going how it was supposed to, we kind of just observed what the tanks were doing. After the treatment with fertilizer, there was an initial algal growth. So on all of the treatment tanks, there was like this film of algae on top. However, over time, that transitioned from that algal growth to duckweed. All 15 of my treatment tanks had a mat of duckweed floating on top, even though all 30 of my tanks were sampled the same way and I used the same tools. None of the control tanks had any duckweed or much primary productivity beyond that. Alrighty, so I did sample some of the tank parameters like temperature, dissolved oxygen, pH, and relative fluorescence. And this was just used to detect the amount of primary productivity within each of the tanks. And these were sampled across 3 days when the sun was at its highest, so around noon. And this is right when productivity would start to really ramp up during the day. So as we can see, pretty much all of the treatment tanks averaged higher than the control tanks. But what we really wanted to focus on was the dissolved oxygen. As we can see, it was a lot higher in the treatment tanks, which was really important for us to see. So a number of weeks after I got the initial sampling, I went out again and sampled all of my tanks throughout the day just to see what the trends were. So some of these make complete sense. Temperatures are going to be lower in the morning, peak in the evening, and then drop off. pH has the same trend. But something that was really interesting to see was that really early in the morning, before any productivity could really have occurred, before there was any sun exposure on the tanks, our control tanks actually had a higher volume of dissolved oxygen. And then in the afternoon, after sun exposure and productivity really kicks up in those tanks, we see that the treatment tanks actually had higher dissolved oxygen. And then it peaks later in the day. And then during the nighttime, this is pretty close to midnight, we can see that the treatment started to level below the control again. So I think this is really important to note because it's likely that there is more consumption of oxygen occurring in those treatment tanks overnight than there is in the control tanks. All right, so getting into the actual breeding activity of the frogs. In total, I got 24 males, and this got me about 167 calling observations. And this mean male activity is just the number of calling males observed per night. As we can see, it's almost a 50-50 split between the control and the treatment tanks. Over here with the female activity, I just used total number of eggs collected because it's the easiest way to determine what the females were doing. They're kind of hard to observe in the field. I got over 6,000 total eggs across this sampling period, and over 90% of those ended up in the treatment. So I think this is a really interesting and really important result to see. And from our chi-square analysis, we can see that the female activity was highly significant. All right, so at the conclusion of the experiment, just, how viable was it? Did it work how we wanted it to? And it really seemed like the manual eutrophication really matched our predictions. We wanted to hit that sweet spot of where The tanks were kicking up in productivity, there was more oxygen, but not to the point where we saw that crash in the system when there was a lack of oxygen killing off everything in the tank. And I think we really got there, and it was really successful for this experiment. Despite some similar measurements in the tank, like temperature and pH, the tanks were visibly different. You could look at two different tanks and tell that one of them had been eutrophied and the other was clear. The eutrophic tanks just had a lot more algae growth and a lot more primary productivity, that duckweed floating on top. And then the control tanks were so clear that you could see to the substrate usually. Unfortunately, during the course of this experiment, there were tadpoles and emerging metamorphs present in many of my tanks, despite my best efforts to make sure all of those were out before I started. So this is just being considered an uncontrolled variation within this experiment. So these were all of my tanks that were noted to have tadpoles or metamorphs. Alrighty, so we saw that indiscriminate choice in males, it was a 50-50 split. It didn't seem like there was any preference one way or the other with the males. And then we saw that highly selective choice in females. Going into this experiment, one of the questions that we had was, would females be selecting for males or would they be selecting for tanks? And I think we actually got the answer to this question, and it's pretty cool. So something that was observed in the field was when there was a female observed in amplexus with a male, there were not actually eggs deposited in that tank. They were in different tanks the next day. So what is possible is that the females are first selecting for males, and then once they initiate amplexus, they actually move to the tank of their choice. So the male choice kind of has nothing to do with it, which I think is really interesting in the case of this experiment. Something else to note is that some males exhibited really high tank fidelity. So that just means that we were finding the same male at the same tank across multiple nights. All right. So this difference in choice really suggests that something is being assessed differently between males and females. So for the males, it's likely that they're assessing predation risk for themselves, or it's likely that it's some type of territorial aspect. So if larger males are choosing tanks and then the other males are just kind of left with like whatever tank is left over, it's kind of hard to say at this point. For females, it's likely that they are assessing the risks for offspring. So things like available dissolved oxygen in those tanks or tadpole predators, because we do know that there are a lot of tadpole predators in many of those tanks. All right, so how I plan to continue with this is to first determine the macroinvertebrate predator abundance between the control and the treatment tank. I do have the samples from all of my tanks. I'm just working through getting all of that processed, and I will have the numbers eventually. The next thing that I'd like to do is increase my egg sample size for the female activity. It's known that Hyla chrysoscelis can have a really large reproductive effort. A single female could drop thousands of eggs herself in one night. So just increasing that sample size this summer would really go to extend my data and just show if this trend is as accurate as we hope it is. Lastly, I would still like to do the tadpole experiment and determine if eutrophication influences larval development, just to make that connection between the parent activity and the larval survival. I think that's going to be a really important part of this experiment to determine, if this behavior is adaptive for eutrophication. So I'd like to thank everybody that's been able to help me through this process, especially my advisor, Cy Mott, for helping me through all of this. Here is my lit cited. And I'll take any questions.